Hey there, Dr. Calloway. Hope these grading these finals ain't been too much of a drag. So today we're gonna get into uh, talking about how all these pathways flow together in our bodies and our cells, and showing how each one is connected to each other, and how when one something happens to one, it ends up affecting all the other ones, kind of like the water change we talked about at the beginning of the semester. So, just to start back at the beginning, where we started at the very beginning of the year, talking about glycolysis and gluconeogenesis. So, glycolysis is the heart of metabolism. It's where glucose ends up getting broken down into pyruvate. And then it's not just there to produce energy because it's not as efficient as a lot of the other pathways that also produce energy. So glycolysis generates 2 ATP molecules per glucose. This gives cells a quick source of energy. Uh, we also see that pyruvate links glycolysis to the TCA cycle, gluconeogenesis, and lactate formation. Once you start looking at the end here at this pyruvate, depending on the body's needs, if we're in a energy deficiency, we'll send that pyruvate back over to gluconeogenesis to convert it back into glucose. Versus if we're in a surplus, like you just ate a big meal or something, have a bunch of glucose and energy now entering your body to be used, we can send it over to the Krebs cycle over here. It goes through the mitochondria and gets converted into acetyl CoA. Then it goes around, um, big old circle there, goes through all those reactions. And then getting back into glycolysis, though. So, uh, one of the ways that glycolysis can kind of feed into other cycles like we had talked about is looking at like this G6P that is up here. So if the body were in a, or if it were someone were to bottleneck there, like if it were to start bottlenecking at G6P, we could start feeding that into the pentose phosphate pathway down here. So we could start feeding into that, so that way the body's still able to use that and just doesn't get completely like bottlenecked and stopped up. Um, and then going into our next pathway from earlier in the semester, we have gluconeogenesis right here, which is just essentially the exact opposite of glycolysis. It's a pathway that allows the liver and kidneys to generate glucose from precursors like lactate, glycerol, amino acids. This process right here is also very important in making sure that our blood sugar levels remain stable, making sure that they don't get too out of whack. And then next, still talking about something from earlier in the semester, be the pentose phosphate pathway. So this branches off from glycolysis, like I had said, through G6P. And there are two main functions that we really look at when we're talking about the pentose phosphate pathway, and that's going to be our NADPH production right here. We use this in anabolic processes like fatty acid synthesis to combat oxidative stress. And then our other um, main thing we look at here is the R5P synthesis. So this is this is really important for the sugars, as they're essential for nucleotide production, and then because of that, linking or just foreshadowing here real quick into something we talked about in the semester would be this R5P synthesis right there is going to get later used when we talk about DNA and RNA synthesis, because that's a very important step whenever we're talking about uh, getting those nucleotides and all that good stuff. So that way our DNA is able to split itself and replicate itself. So next, talking about the TCA cycle real quick. So the TCA cycle starts after we have that pyruvate from glycolysis sent, goes through the mitochondria, goes into acetyl-CoA, and then whenever we start looking at the energy production from this, we can start looking at the NADH and FADH2 that it produces. So start looking at these junctions right there. And these are important because these are electron carriers that transfer energy from the ETC, which is generating ATP. And then it's also this cycle can also act as an intermediate for alpha ketoglutarate and oxaloacetate, as these are both crucial whenever we start talking about amino acid synthesis. So talking about those proteins. And then the TCA cycle is also super important because it essentially just connects almost every single pathway. You're able to link it there some way or another. So like for instance, acetyl-CoA links to the fatty acid synthesis, as well as fumarate connects to the urea cycle, which I have drawn over here. So like looking here, you have fumarate down there. And then next, start thinking about electron transport chain, sorry. And then oxidative phosphorylation. So this is where that NADH and FADH2 start donating their high energy electrons. So start looking at here, you have these electrons, they go into the complex one, it starts bouncing around, it can't hold it, it wants to get rid of it, and then it makes its way through all these different complexes, or our CoQ right there, and essentially ends up here in our ATP synthase, which is what's really starts actually producing a majority of the ATP that we see through the electron transport chain. So whenever we start thinking about oxidative phosphorylation, ETC uses oxygen to produce a large amount of ATP, making it the most efficient energy generator, which is why that's why a normal cell uses it to produce all of its energy, because it's the most efficient way to do it. Versus we start thinking about something like in question 3 with the cancer cells, where those actually make their energy through glycolysis, which is a lot more inefficient than it is to make it through ETC. And that's part why they're a problem. So next, moving on to amino acid metabolism. So amino acids serve as both building blocks for protein and intermediates in metabolism. So we start talking about catabolism of amino acids, such as those like alanine and glutamate. Those both feed into our TCA cycle. So we have our alanine, and then they feed into the TCA cycle, and they go through a glucogenic amino acids, which are contributed to glucose production. Well, ketogenic amino acids like leucine generate acetyl-CoA for energy. So have this see it start producing energy, or start generating acetyl-CoA for energy, sorry. And then start talking about anabolism, metabolism, and we start looking at how the TCA cycle supplies the precursors for amino acid synthesis. So, for example, oxaloacetate forms aspartate, and alpha ketoglutarate forms glutamate. And then the nitrogen, going back to the catabolism, the nitrogen released during catabolism ends up getting managed through the urea cycle, which we'll end up discussing here in a bit. So next into fatty acid synthesis and breakdown. So from acetyl-CoA, we get derived, or the acetyl-CoA, which ends up getting derived from amino acids, ends up being our starting point. So we have the malonyl-CoA formation. So this is the enzyme acetyl-CoA carboxylases catalyzes, which is the first step. Sorry, that was a mouthful trying to get out. And then we have our NADPH dependence, which is our fatty acid synthase, which uses NADPH from the pentose phosphate pathway to elongate chains like palminate. And then we start seeing that these fatty acids break down via beta oxidation to produce acetyl-CoA, which again feeds back into the TCA cycle, showing that the TCA cycle is really connected to a lot of these different pathways. And then this ends up helping us ensure lipid and carbohydrate metabolism remain balanced. And then we have nucleotide synthesis and degradation. So, the pentose phosphate pathway produces that R5P like we were talking about before, which is important for our nucleotide synthesis and degradation, and ends up getting us our two different parts of it, which are our purines and our pyrimidines. And then our purines end up going to be built from amino acids like our glycine and glutamine, and then our pyrimidines are going to be synthesized through aspartate and carbonyl phosphate, which ends up connecting us back to the urea cycle. Which leads us to last tidbit here, which ends up being the urea cycle. So this is how the body manages nitrogen. This is the way we get rid of nitrogen. It's through the urea cycle. So the ammonia, which is produced from our amino acid catabolism, is really toxic for us, and we need to excrete it. Otherwise, it won't be good. So the urea cycle is what helps us convert that ammonia into urea, which we are then able to safely excrete via the kidneys and eventually as urine. So we start thinking about how the urea cycle interconnects to everything else. So like we talked about, we have the carboxyl phosphate, which feeds in here, and then we start thinking about other things like fumarate, which end up coming out of the urea cycle and go back to feed to the TCA cycle, which is back there at the top. So this helps ensure the efficient use of carbon skeletons when detoxifying nitrogen. So this is making sure that we aren't just wasting carbon, and that we're also at the same time making sure that we are getting that toxic uh, ammonia out of us and getting that, or excreting that extra nitrogen. So uh, essentially, big picture, the urea cycle is important for, again, helping us keep that nitrogen balance in our body and not allowing a toxic buildup, and also just being a balancing act between that flow of nitrogen and our carbon metabolism. So, uh, just getting into big picture things here to end us off at. So, when we just start talking about all these pathways and how they're all connected, uh, we can start looking at like the carbon flow. So, like how glucose from glycolysis feeds back into the TCA cycle, fatty acid synthesis, amino acid metabolism, and helps just intermediate um, between those pathways, and helps us ensure flexibility so that we don't get too bottlenecked at one location and can start using that energy efficiently in others. And then next, we start thinking about how the nitrogen flow between all these pathways is integrated. The amino acids supply nitrogen for our nucleotide synthesis and our urea cycle, which help keeps our system balanced, helps us from keeping too big of a buildup of nitrogen. And then uh, next would be just thinking about general energy and our redox balance. So like ATP and NADPH generate glycolysis, the PPP pathway, and OXPHOS. So these all help produce energy for the body by converting it into usable uh, things for the next pathway to be able to more efficiently do it. So, like we had talked about, having glycolysis feed into Krebs, which feeds into the ETC, and then lastly, just wanting to uh, touch up on more of our DNA synthesis and the making of that. So we start here on the left. We'll see this is our DNA, like our double helix right there. And then we'll have our helicase coming through. It's basically just unzipping that DNA strand, turning it into RNA, where we'll eventually have mRNA coming through. It's uh, reading these proteins that are on it, and it's doing that via looking at these codons we have here. So for something like AUG, that's our start codon. It's looking here at the 3' end, and it's going to work its way down to our 5' end until it comes into contact with the stop codon, which is UAG. So starting here, it's going to give us the complementary nucleotides. So U, A, and then U, and it's going to work its way down, replicating that DNA for us to be wound back up into its double helix. So looking more, we can see we have our tRNA right here, which is, sorry about that. We have our tRNA here, which is taking these amino acids, these chains right here, and converting that into these nucleotides, these proteins right here that end up connecting to our strands on our RNA. And then looking at here, we have just our uh, some of them disaccharides we talked about, as long as some of them longer polymers. I did not have enough time to draw these out, but I put the names there. So, and then just looking at all these amino acids we have up here as well, all that stuff, or all the amino acids play part into this. But yeah, so just concluding thoughts, those are how all these biochemical pathways are integrated, and just kind of walking through them again. And then, yeah, so just thinking about how they're all integrated right there. I think I went into enough detail for each one, hopefully. Um, Sorry, this is not my best work right here, Dr. Calloway. I was working on it last night in my bed, and I woke up at about 8 this morning and had a computer and an iPad in my lap. So I'm kind of scrambling here, trying to get this submitted on time. So, hope you enjoyed it. Hope you have a good Christmas. Sure, I'll see you again before you get this graded, but if not, then I hope you have a good Christmas.